Okay, and welcome to today's Wednesday afternoon lecture. I'm Chris McBain, the Scientific Director of NICHD, and I'm thrilled to introduce today's wall speaker, Dr. Gisela, Gigi to us, Storz, who's made remarkable strides in her research on small regulatory RNAs and small regulatory proteins. Before I do introduction, I have two quick announcements. The first is the code for continuing medical education credit. The code is 66817. I'll repeat that, 66817. And also, for those of you who are watching remotely, you can submit questions to the speaker anytime during the lecture. Simply click on the button that says Send Live Feedback, which is below the videocast window. Type in your message and it will be relayed to the speaker at the end of the lecture. Okay, now to our guest of honor. Dr. Storz received her PhD from the University of California, Berkeley, where she trained with Dr. Bruce Ames. She first came to NIH as a postdoctoral fellow with Dr. Sankar Adya in 1989, and then was recruited to an independent position in cell biology and metabolism branch of NICHD by Dr. Rick Klausner in 1991. One of her major research interests has been the identification of genes that have long been ignored. First, genes encoding regulatory RNAs, and more recently, genes encoding the smallest proteins, which also act as regulators. Having discovered many of these missing gene products, her group has gone on to reveal their mechanism of action and key physiological roles. Dr. Storz has served on several NIH committees, including the Central Tenure Committee, the Committee on Scientific Conduct and Ethics, and is now serving as the chair of the Women Scientists Advisors Committee. Among many awards, Dr. Storz has received 5 NIH Directors Awards, including the Ruth L. Kirstein Mentoring Award, and was elected member of the National Academy of Sciences in 2012, and was elected to the American Association for the Advancement of Science in 2024. She's currently an NIH Distinguished Investigator and head of the section on environmental gene regulation. On a personal note, I think Gigi represents everything that is great about the NIH intramural program. She is doing remarkable, innovative, cutting-edge research, but at the same time acting as a mentor not only to trainees in her lab, trainees inside our entire IC as well as across the NIH. She's also on too many committees and too many mentoring committees for faculty. I don't know how she has the bandwidth to do half of it, but I'm glad she does. We all benefit from colleagues like Gigi. So I'm proud to say that she's a member of the NICHD community. And with that, I'd like to recognize and welcome Dr. Storz, who's going to give us a talk entitled A New Universe of Regulatory Microproteins. Over to you, Gigi.
Thank you, Chris, for that very kind introduction. I feel lucky to be in NICHD. So let me get right into my talk. A great deal is known about the proteome now in all organisms. So we know about enzymatic activities, we know how proteins act as regulators or have structural roles. The actual structures of millions of proteins have been solved, and we're quite good at even predicting the structures of many proteins at high accuracy. But despite all we've learned, there's a whole class of proteins that have been ignored. And these are the smallest proteins, shown in red here. Sometimes they're referred to as the dark proteome. And I'm referring to proteins that are of around 5 kilodaltons or less. So they just have been ignored for multiple reasons that I will mention. As someone who has been long interested in genes that have been missed and the roles of these gene products encoded by the genes that have been ignored, I was really fascinated by these small proteins. And I think it's important that we're aware of them and learn more about their functions, because as more and more are characterized, it's becoming clear that they act as regulators of larger proteins by binding to these regulators, by binding to these proteins and modulating their functions or modulating their stabilities or activities or localization. Before I proceed, I should say a little bit about the nomenclature, which is somewhat sort of squishy, in part because I think this is an emerging field. So what comprises the dark proteome? The upper size limit is a little bit arbitrary. In bacteria, historically, we've focused on proteins of less than 50 amino acids. And we've called these proteins small proteins. The reason for this 50 amino acid cutoff is that's where annotation traditionally stopped. So when bacterial genomes were annotated, proteins of less than 50 or sometimes 40 amino acids just weren't annotated. And I'll comment on that a little bit more later. In eukaryotes, let me see if I can get this to work. Oh, there it is. There it is. Okay. So in eukaryotes, the cutoff for annotation more commonly was around 100 amino acids. And here the term microproteins is more commonly used. So in my talk today, I'll talk about both small proteins and microproteins interchangeably, but I'm referring to these smallest proteins. Sometimes they're also called SEPs, small orphan-coded proteins. So here I am focused on proteins that are encoded as a distinct entity. So there is a start codon and a stop codon. So this is encoded as a defined gene. These can be encoded in a variety of different places. So there's one group of proteins, or some of them are encoded between other genes in intergenic regions. They can also be encoded internal to longer open reading frames. So the genes encoding these longer proteins can have genes within them. Sometimes these are called alt-orfs or genes within genes. These can be encoded in the same frame as the longer protein so that they share a stop codon, but they can also be encoded out of frame so that the start and stop are out of frame to the longer protein. They're also encoded by regulatory RNAs. This is one reason I don't like the term non-coding RNA. For example, long non-coding RNAs or linked RNAs are often thought to just be regulatory RNAs, but a fair fraction of these have been shown to encode a small protein. So I want to distinguish these groups of proteins from peptides that are cleaved out of a longer protein, either through a very defined process or throughout the degradation of the protein. These peptides, I think, can also have important functions, and I think are also being missed. I suspect there's a lot more peptides or products from proteolysis that could have independent functions. But today I will focus on the small proteins or microproteins that are encoded by a defined gene. So why has this dark proteome been missed? So as I already alluded to, a big problem is this issue of annotation, which I've tried to illustrate here. So if you look at a region of the genome, there can be many potential open reading frames indicated here by random green start codons with the green arrows or random red stop codons with the red arrow. So just in any region, there's a high potential to encode an ORF, or there to be an ORF encoded of, say, 8 or 10 or 15 or 30 amino acids. And it's very difficult computationally to distinguish which of these small ORFs are in fact translated and which ones are just there by chance. So related to this problem of annotation, this also impacts sort of genetic approaches. So people may identify mutations in of interest in a certain region, and they may not realize that this is affecting a protein that is encoded here if that is not annotated. So a mutation may be misinterpreted as possibly having a regulatory consequence for the expression of a neighboring gene, when in fact it affects the expression of a protein encoded here. The genetic problem is also exacerbated by the fact that these genes are small, so it's difficult to hit them in insertion mutagenesis. And also related to the issue of annotation, if groups are carrying out CRISPR screens or SI screens, if something is not annotated, you will not have knockouts or knockdowns of these genes. So probably the biggest problem, though, is that many biochemical approaches are not optimized, or they're optimized for larger proteins. Sometimes it can be very difficult to detect the smallest proteins, so they may behave differently in gels or other matrices used to purify or to analyze proteins. Just as one example, standard approaches for detecting proteins, larger proteins in gels, such as Comasi blue staining and silver staining, rely on there being certain amino acids in the protein so they can be detected. But if your protein is only 20 amino acids and doesn't have that particular amino acid or only one of them, you may not detect it. So as an example of the challenges in detecting small proteins, I just want to mention work carried out by Aoshu Zhang in my group. He has been characterizing DNA endonuclease proteins referred to as RPN proteins. So these RPN proteins were characterized by New England biolabs, from which we get a lot of enzymes that act on DNA. So NEB is interested and purifies a lot of proteins that act on DNA, and they also characterize some RPN proteins. But they were not very interested in these RPN proteins because they found that the DNA endonuclease activity was not very strong. They just noticed that the activity wasn't very strong. And the reason is, as Aoshu showed, is that there is an alt-orf encoding a small protein within the RPN gene, and this small protein blocks the activity of the longer protein. So NEB purified their RPN endonucleases and didn't realize that there was another protein inhibiting the activity of the longer protein. So when Aoshu purified both the long protein and the short protein, he could document how, in fact, if you just had the long protein, it was good at cutting DNA, while the mixed population purified by NEB really was not as good at cleaving DNA. And you could inhibit the activity of the longer protein by just adding the short protein. And this has made me wonder how many other purified proteins people have isolated, not knowing there might be another protein associated with this larger complex, which could be affecting the activity. So, as I mentioned, we've been interested in identifying the genes encoding microproteins or small proteins, and then characterizing the functions of the proteins. So the first small proteins were identified by serendipity, some phenotype associated with overexpression of a piece of DNA. But what has really made a difference more recently is ribosome profiling. And I want to say just a little bit about ribosome profiling. What is ribosome profiling? This is an approach in which you isolate translating ribosomes. So you isolate polysomes and the fraction with translating ribosomes. The extra RNA that is not covered by the ribosomes is digested away. So you isolate just this fraction of RNA, which is converted into cDNA, so that you can determine by deep sequencing what regions of RNA were bound by ribosomes. So there are many permutations of ribosome profiling. One permutation that's been very valuable to us that I want to mention is that you can add antibiotics to your cells at the outset and lock ribosomes on either start codons by certain kinds of antibiotics or lock them on stop codons, which helps delineate what the ends of a gene are, what is being translated. You can identify start codons and stop codons. And this has been a really valuable tool in identifying the very small ORFs that are being translated. So let me show you an example of the data. So this is a gene in E. coli, and without any antibiotic treatment, you see ribosome profiling signal across this whole coding sequence, whereas in the presence of these antibiotics, either UNC112, the signal is in blue, or retapabulin, the signal is in red, you see peaks just at the start codon. The work on this project was carried out by Jeremy Weaver in my group, and we collaborated with Alan Buskirk, who generated the UNC112 data, and we used data from the Vasquez, Laslop, and Mencken groups. The two groups use slightly different ribosome profiling protocols, which is why these two peaks are not on top of each other. But you'll see other examples of this, this sort of ribosome profiling data. But this was enormously useful in helping us identify new small protein or microprotein encoding genes in intergenic regions. So just in E. coli, if you look at open reading frames between 8 and 50 codons, just in intergenic regions, there's nearly 200,000 possible ORFs, illustrating the problem with identifying the real coding sequences. But now, with the ribosome profiling data, we could see where you had strong signal near a start codon as opposed to sort of more random signal. And so we could see what open reading frames were the best candidates for encoding small proteins. And what we always do when we think we have an open reading frame that encodes a protein is we tag it on the chromosome. We introduce a tag just upstream of the stop codon. And as shown by the many blots here, this approach was quite successful. Those that had very strong ribosome profiling signal, in fact, could be detected as proteins, 38 of the 41 that were tested. As we are learning more about the microproteins or small proteins, the computational approaches are catching up, and in a collaboration with Igor Fesenko and Eugene Koonin, we also looked at proteins that were identified computationally. And Eugene Koonin's group took advantage of the fact that there's many enterobacterial genomes that are very closely related in sequence. So they could mine these genomes for intergenic regions that had some evidence of conservation, of a difference in the ratio of non-synonymous substitutions versus synonymous substitutions. So with this large amount of sequencing data, you could look for these signatures of positive selection. Here again, in this case, Rajat Dahani and my group tested several of these candidates for expression in E. coli and detected 11 of the 16 candidates that he tested. So this leads me to what, this introduction leads me to what I'm going to focus on today, and that is, what do these small proteins do? And the two take-home messages I hope I can convey are there are thousands of microproteins to be discovered. We're missing many, many of these proteins, and that they serve as regulators in all domains of life. And what I'd like to do in the rest of my presentation now is to describe different examples of how these small proteins act. And although we have focused on bacterial cells, I think it's hard to convey how difficult it is to work on these small proteins. There's a reason people didn't work on them. But working with bacteria, we can carry out many different types of approaches very rapidly, so we've been able to make some progress. But I've chosen examples of ways the small proteins or microproteins can act, which I think have been shown in other organisms in eukaryotes and are likely to be, having similar roles in other organisms, or their similar functions are likely to occur in other organisms. So I will describe 4 examples. The first of these is a 15 amino acid protein that binds to a transcription factor and represses the activity of the transcription factor. I'm starting with this example because it illustrates how we sort of segued into the small protein area of research through our study of small regulatory RNAs, because this small protein is encoded by a regulatory RNA. It is encoded by the SPOT42 RNA, and the expression of this 15 amino acid protein is highest when cells are growing in glucose. So SPOT42 was actually one of the first small transcripts to be characterized in the 70s. It was identified as a phosphate labeled spot on a 2D gel when people were looking for tRNAs. So it was a defined spot, and then later shown to be a regulatory RNA. In bacteria, most of the regulatory RNAs act by limited base pairing with one or more targets encoded elsewhere, sort of through limited complementarity, analogous to how microRNAs act in eukaryotic cells. So it was known that expression of this RNA is highest in glucose, and that's also shown on the northern blot here. It was also shown that expression is repressed by a global transcription factor called CRP. So CRP represses transcription of the SPOT42 RNA, and it turns on the expression of a lot of genes required for the utilization of non-preferred carbon sources. Interestingly, these same genes that are activated by CRP are repressed by SPOT42 when cells are grown in glucose. So this regulatory scheme is shown here. E. coli cells prefer glucose over everything else. So if they're growing in glucose, they want to make sure that the genes for enzymes or transporters for non-preferred carbon sources are turned off. And this is what SPOT42 does. It base pairs with these and makes sure they're off. When cells encounter low glucose, you now repress SPOT42, and now the CRP transcription factor turns on these genes for the use of alternative carbon sources. So we thought we knew everything there was about SPOT42. There are many targets. We characterized the targets. And given this, we were really surprised to see a strong ribosome profiling signal in the middle of this regulatory RNA. So this suggested that, in fact, this regulatory RNA was also translated. We looked and could see there was a possible ribosome binding site and a start codon, followed by an in-frame stop codon, which could lead to this pretty nondescript 15 amino acid protein. So I wanted to see, or we wanted to see, is this actually translated to give the protein? So we tagged it like we often do, and could see that the protein was in fact synthesized. The 15 amino acid protein was synthesized, as was expected. The levels were highest in glucose, but we also noted that the levels increase with higher temperature, likely because we think the ribosome binding site becomes more accessible as the RNA structure unfolds. So we have an RNA that acts by base pairing, and overlapping the sequences required for base pairing, we have a protein coding sequence, as is shown here. So here we have the protein coding sequence. These are two regions involved in base pairing when spot 42 is acting as a regulatory RNA. We wanted to know, and I should say this work was carried out by Jordan Aoyama and Meta Reina. They wanted to know, does the protein have function? And when you have overlapping coding potential like this, it's a little bit tricky to deconvolute the two possible functions. But what they did was to take this sequence and recode it so that you would still make the same protein while eliminating the base pairing potential. So now we had a transcript that would only act as an mRNA. They also made a version where they introduced a stop codon into the coding region, leaving the sequences involved in base pairing. So now there were products or versions of the RNA which would only act by either base pairing or by encoding a protein. And interestingly, the two different constructs, when we made both of these derivatives, they had the same phenotype. They both blocked growth on galactose because they inhibited those enzymes required for galactose utilization. So they act in the same pathway and they have the same effect. And this also showed that both the protein and the RNA have regulatory functions in the same pathway. But for all the small proteins, what we always are looking for is what else do they interact with? And when you have a protein that's only 15 amino acids, that can be kind of challenging because when you add tags to it, you could affect the activity of the protein. So what we did for this 15 amino acid protein, which we call SPFP, we introduced a stop codon, which could be recoded by to insert a non-native amino acid just downstream of the start codon. So by introducing the stop codon and adding this specialized amino acid, we could biotinylate the protein and pull it down and purify it. And we were really intrigued to find that one of the proteins that co-purified with the SPFP protein is the CRP transcription factor I have mentioned multiple times, which is involved in the regulation. With all these proteins, we always carry out reciprocal purifications to convince ourselves that there is, in fact, an association between the two proteins. So if we pull down CRP, we can also detect the 15 amino acid protein. And what's more important is that it could affect the activity of the CRP transcription factor. So these blots, these Western blots, show 4 proteins whose expression is increased by CRP in either galactose or in maltose. And if you look at these middle lanes, when you overproduce the 15 amino acid protein, you no longer get good induction of these different examples. So the 15 amino acid protein, by binding to the transcription factor, is blocking its activity. It's inhibiting it. So that allowed us to add to our model here that spot for the SPFP protein acts in the same pathway, ultimately also blocking expression of genes for non-preferred carbon sources. But in this case, SPFP interacts with the CRP transcription factor. We think at lower temperatures, the base pairing RNA works very well. It acts as a good repressor. It doesn't work as well at higher temperatures. At these higher temperatures, the protein is translated, but it has the same regulatory effect. But I think the most important point I want to make with this example is I wonder how many other transcription factors are out there in bacterial cells and eukaryotic cells whose activity is modulated by such a small protein. So CRP was studied by many, many different labs. Many different structures were determined for CRP, but no one ever noted this small protein associated with CRP. So, and in bacteria, there are other examples of proteins of roughly the same size, 10 amino acids, 12 amino acids, that are generated as peptides and are released by phage or released by certain bacteria at certain densities, which also have similarly been found to bind to and modulate the activities of transcription factors. So if there's some cavity in a transcription factor, one can easily imagine how a small protein of just 15 amino acids, 10 amino acids could bind there and modulate the activity. So now let me move to the membrane, where the majority of small proteins or microproteins characterized thus far have been found to act. The first of these examples is a 28 amino acid protein that is an amphipathic helix and recruits enzymes to the membrane. So this was another case where we were characterizing what we thought was a regulatory RNA and found that it encoded a protein. So it overlaps a base pairing RNA. Again, the expression is regulated in response to carbon availability. It's also strongly induced at low pH. So this was another case where we saw a strong ribosome profiling signal on what we thought was a regulatory RNA. When we looked at the 28 amino acid sequence encoded here, we could tell it was an alpha helix, and in particular, it was an amphipathic helix, which we thought might associate with a membrane. So one half was hydrophobic, the other half had charged residues. And when we tagged this protein we call Azuc with GFP, we saw, in fact, that it was associated with the membrane in bacteria. And we could mutate residues we thought were important for the association and no longer saw, well, we lost the association. So we thought Azuc, or these data suggested to us that Azuc associates with a membrane. Once again, we wanted to know what proteins the amphipathic helix is binding to, and found that it binds to a glycerol phosphate dehydrogenase. So we could either purify based on the glycerol dehydrogenase, GLPD, or purify based on the small protein, and we saw co-association in both cases. Did it impact glycerol dehydrogenase activity. In fact, it did. It had been previously reported that this enzyme activity was stimulated by amphipaths, and sort of consistent with that, if we just took cell extracts, again, this was work by Jordan Aoyama and Medarena, we saw that extracts from cells lacking the small protein had lower activity than the wild-type cells. And if we overproduce the 28 amino acid protein, we saw elevated activity compared to the vector control. So this amphipathic helix protein is modulating the activity of the dehydrogenase, and we think it's through recruiting the dehydrogenase to the membrane. And this shows the localization of the dehydrogenase GLPD fused to YFP. In the presence of the small protein in a wild-type cell, you see more association with the membrane compared to cells lacking the protein. Again, I think this will be a very general mechanism by which proteins could be recruited to the membrane under very specific conditions. So Zook is induced at low pH. And one reason I say that is that a set of proteins being characterized by Joe Schmidt in the group is suggesting the same thing for another dehydrogenase and another amphipathic helix protein. So this is one of the examples of a gene within a gene, or an alt-orf. So the GND, the large gene, encodes a phosphogluconate dehydrogenase, and encoded out of frame with different start and stop codons, there's an amphipathic helix protein. I should mention this was first noted by Sarah Slavoff at Yale University. But we're intrigued by the fact that, again, you have a dehydrogenase and an amphipathic helix. I think it's important to document these proteins are made. We see the full-length protein under all conditions tested here. Interestingly, the small protein is specifically induced under conditions of heat shock, under aerobic conditions, and you can't detect it under anaerobic conditions. So the amphipathic helix protein is made under very specific conditions, and so we wonder, under these heat shock conditions, it might also be important to bring the dehydrogenase to the membrane. Before I end with this example, I also want to mention the work of Kumaran Ramamurthy, who's really beautifully shown yet another amphipathic helix protein, the SPO5M protein. Again, similar size, amphipathic. In this case, this protein specifically associates with membranes of a certain curvature and forms the basis of the proteins that assemble for the formation of a spore in Bacillus subtilis. So we have a small protein that binds to a transcription factor. We have another one that recruits enzymes or other proteins to the membrane. I now, in the last 10 minutes, want to turn to proteins that are found as a transmembrane alpha helix, just a single transmembrane alpha helix to regulate the activities of transporter proteins. The first of these is larger than most of the proteins we characterized, and this is probably why it was easier to characterize. The expression of this protein, the 49 amino acid protein, is induced in response to various antibiotics or other noxious chemicals such as oxidative stress. So when we purified this ACRZ protein, we found that it associated with a component of a multi-drug efflux pump, the R&D efflux pump. And again, we could purify the R&D efflux pump, the ACRB component, and get co-association of the small protein. So these R&D efflux pumps are one of the reasons bacterial cells are resistant to so many different kinds of drugs. They can export a whole variety of different drugs, here indicated by purple dots and red dots. So the ACRB component is in the inner membrane. It's connected to TOL-C, which is in the outer membrane, through the ACRA protein. And these pumps act to transport these compounds out of the cell, sort of preventing damage by these different drugs or noxious chemicals. It's known ACRB mutants, cells lacking the efflux pump, are sensitive to a whole variety of different antibiotics shown here. These are competition assays between wild-type cells and cells lacking the ACRB inner membrane component of the transporter. What we found is that cells lacking ACRZ are also sensitive to some of the compounds, but not all of them. So they showed almost wild-type resistance to things like erythromycin, but were very sensitive to chloramphenicol. So that led to the model. Ooh, sorry, this is probably a big slide, yeah. The model shown here, that the ACRAB TOL-C pump is really good at pumping out the red compound, but is not as good at pumping out the purple compound unless there's also the small protein, which allows this pump to become a better transporter of the purple compound. So how could this be? How could binding of this small protein increase efflux of the purple compound? Well, in a very nice collaboration with Ben Louisi's lab, where they obtained a cryo-EM structure of both ACRZ together with the whole complex, they found that binding of the small protein actually leads to allosteric changes further away in drug binding pockets. So this interaction of ACRZ with ACRB is changing the structure, which we think in some ways impacts the binding of certain kinds of compounds. Interestingly, this effect of ACRZ is synergistic with cardiolipin. So there's even more specificity in the presence of cardiolipin. And I think that likely will be the case with other small proteins, that they will be synergistic with the lipid environment. There are many, many efflux pumps. And they're the reason a lot of bacteria are resistant to many different antibiotics. For example, this is a pump in Pseudomonas. This is a pump in Neisseria. Both of these are potential pathogens. And I find it intriguing that in a position where we find ACRZ, you see sort of a cavity. So you wonder if these proteins may also have a transmembrane alpha helix protein that can modulate their specificity under certain conditions. There's even R&D efflux pumps in eukaryotic cells. Here are the structure of the Niemann-Pick type C protein. And again, one could imagine how they could be modulated by a small protein. So now let me turn to the final example. This is a 31 amino acid protein, also a single transmembrane alpha helix. And here a big clue came from the observation that the levels of this protein are highest when cells encounter low magnesium concentrations. And data showing this is on this slide. Here we look at the levels of the mRNA. You can see without magnesium, the levels are highest. The same is true for the protein. So that was already a big clue. We thought that the protein was acting under conditions where there was low magnesium. So when we did the co-purification assay and saw co-association with the magnesium imported, we thought this might be the likely target, and this was confirmed by reciprocal purification experiments. So MGTS, the 31 amino acid protein, binds to the magnesium importer. In this case, it impacts the levels, and we think the stability of the importer. So if you compare cells, wild-type cells with cells lacking the small protein, you see far higher levels of the MGTA protein in the wild-type cells. In contrast, if you overproduce the small protein, you see higher levels. So here, the small protein is impacting probably the accessibility to a protease. We, of course, were very interested in seeing this interaction between MGTS and MGTA and had a great collaboration with Doreen Matthews and Alex Zolt in NICHD. Unfortunately, the structure that we resolved, or that Riley Zeiner, the postdoctoral fellow working on this, was just the large protein. We saw a very nice dimer and learned a lot about MGTA. But this, we had to model in the small protein via AlphaFold, and don't think this prediction is very good. But we at least learned about the larger protein. Zachary Rich, a graduate student in the lab, is now going to get this co-structure. But we're very interested in this whole family of proteins because these are all P-type ATPases, of which there are many in all organisms, and they transport a whole variety of different ions from calcium to magnesium and zinc. This diagram was generated by L. Ervand and NLM. So this is where we see MGTA. It's really quite related to calcium transporters found in eukaryotic cells, the Circa pumps, which are found, for example, in muscle cells where they transport calcium. And the reason we are so interested in this is one example in eukaryotic cells where other microproteins or small proteins have been found to regulate these proteins, these transporters. And these are actual structures that have been determined for the Circa pump, this calcium transporter, which is defective in some diseases, and its association with two different microproteins. So it makes us wonder how many more of these different types of transporters are also regulated by small proteins to affect their activities, their specificities. And I've just told you about two types of transporters, P-type ATPases and R&D efflux pumps. But there are many, many different types of transporters, for example, in nerve cells or in kidney cells, important transporters. How many of those may also have a protein that regulates their activity, just a single transmembrane alpha helix that alters their activity under a specific condition? And one reason I say that is we're intrigued by another small protein identified by Igor Fesenko together with Eugene Koonin that is encoded adjacent to another transporter, a member of the calcium transporter Bax inhibitor family. Here, Igor predicted that this small protein might, in fact, interact with this Bax inhibitor family protein, and we found that if you overproduce the small protein, in fact, the levels of this transporter protein go down. So with all the different membrane proteins, particularly transporters that are present in so many different kinds of cells, how many more of these are modulated by a protein that we haven't yet detected? So let me go back to my take-home message. I hope I've been able to convince you that we're missing this whole dark proteome, these very small proteins, but that they can serve as regulators, and I think they will be important in all domains of life. What I find most exciting is the questions which we really haven't addressed at all or just beginning to think about. If proteins are regulators, their activities or levels also should be regulated. How does that occur for microproteins? How do these proteins evolve? And ultimately, can we exploit them? Something that's 15 amino acids is easy to synthesize, so 31 amino acids. Can we exploit these proteins? Now I'm going to turn to my most important slide and want to acknowledge the members of my group. I've tried to say who did the work throughout my talk. This is my current group. These are former lab members who've contributed to the work that I talked about today. I should say I've now been at NIH for almost 35 years and feel really fortunate by the great group of people that I think have done great science and have been great fun in my group. So I really appreciate that. I just also want to acknowledge that we have had terrific collaborations with people around the world and particularly also in the intramural program. And it's my understanding that Dr. Mider was very supportive of the intramural program, particularly NLM. Here he's, I think, giving the award to Maxine Singer, who was really supportive when I first came to NIH. So on the occasion of this talk, I decided I would look up how many people I've published with here at NIH. And I've really published with people from all kinds of institutes. And I just want to end by expressing my appreciation. This is just the people with whom I've published papers, but many more people have given advice to my lab, provided critical feedback or reagents, let us use a piece of equipment. And I've always thought the best aspect of the intramural program is the wonderful colleagues that I have here. So I want to express my appreciation for that and end there. Thank you for your attention, and I'm happy to answer any questions.
Folks know where the mics are. Please use the mics. We do have a question online to start things off. You're on. Okay. Very neat, Gigi. You may have mentioned it, but do these little proteins have promoters?
Yes. So some are encoded as part of longer messages, as part of an operon, but many are encoded as a defined transcript with their own promoter that controls their expression. Okay, Harris.
Yeah, hi, Gigi. That was great. I have two questions. One, I think I asked you about a decade ago, but I don't know if anything has been learned since then. Is anything known about how proteins like ACRZ are integrated into the membrane? And the second question is, what about small proteins in 5' untranslated regions like TNAC that Charles Janowski discovered? Are there many other examples of that in bacteria or other organisms?
Yes. So sorry that we haven't been able to make more progress in answering your question. We did do one study where we depleted components of the secretion apparatus, and they have stronger effects for some proteins than for other proteins. To be honest, another group has published a paper saying that maybe we misinterpreted our results. The catch is we have to use tag proteins to look at the transport to the membrane, and it's possible that the tags are having an impact. So I don't think we fully understand how they get into the membrane. I think that's an important question. So UORFs, or leader peptides, I think that's a super interesting area. The work with Eugene Koonen's group indicates there's a lot more of these than we ever imagined. And Maciek in my group, is one of his real interests is, how many of these UORFs have independent functions? Do they act in a regulatory capacity in cis, impacting the downstream gene, and also have a separate function? But none of the ones that I talked about here are those. The trickiest part is documenting that they have function.
Okay, thanks.
Okay.
Heike Yee, very nice talk. Thank you. This is possibly a somewhat naive question, but obviously, as you say, in bacteria, you can identify these proteins more easily. Has anybody looked to see if they exist in eukaryotes as well, or would you expect them to be altered? Is that a fruitful approach?
So there are now databases of thousands of these in mice and human, where people see ribosome profiling signal or sometimes computational evidence. The catch is only a limited number have been documented to actually exist in cells because it's harder to tag proteins, and for even fewer is there a known function. But there are now catalogs of thousands of these kind of proteins, of small proteins, less than 100 amino acids in eukaryotic cells.
We can talk later. Thank you.
Yeah.
Thank you for a really interesting talk. I think that your work with the small proteins influencing the efflux transporter specificity is really fascinating. Is there a way that you could find these small protein regulators by looking at the protein that you're interested in? I know that you saw those interesting kind of grooves of binding sites. Is there any way that you could predict from that, or how would you go about doing that?
We would love to do that experiment. So Mona Orr, who worked on the R&D efflux pump regulator, she's tried to do that for Pseudomonas and has not been able to find a protein. And I think it's for technical reasons. So I think we need to optimize how to do cross-linking perhaps. Mass spec for proteins this small is also not very good at all. So we need to work on techniques to allow us to do that. I think that would be a major breakthrough if we could take large proteins, sort of your transporter of interest, under a number of different conditions, purify it and see if you see an associated small protein. So I would love to do that.
That would be really cool. Thank you.
Hi. Great talk. For the small proteins that affect the transporters, is there any way to cut them off and see how many generations it takes for the effect to go away?
Well, that's an interesting experiment. We have not done that. It sort of gets at the question of how is it turned off, about which we know very little. We haven't done that experiment.
And any sense of the in vivo stoichiometry between, is it like 100 to 1? Or?
That would be great to do. Because we don't have antibodies, it's technically far more difficult than you think it would be. Mm-hmm. So we do not know that, yeah.
Thank you.
But it's an important question. Anirban.
Gigi, that was beautiful as usual. So I have two perhaps naive questions. The first one is that you showed Alan's data, the data from the other group where the ribosome profiling was stopped slightly different ways, and you got different results, right? And for all of these, for example, for something like ribosome profiling, the biochemistry has pretty much been standardized. And my question is that, are there ways to do these things differently, and could that then lead to discovery of more different small proteins, like it seemed that those two data, they actually led to different small proteins, but correct me if I'm not.
Yeah, so I didn't want to go into the details. So the ribosome profiling can be done with sort of nucleases, different nucleases digesting the RNA, some digest in further, and the two labs use sort of different nucleases or different lengths of time of the digestion. That's the reason why the peak is shifted. It's a technical reason. But yes, there are ways, I think, to optimize ribosome profiling. So, for example, some groups are now combining the approach of stopping or trapping the ribosomes on start codons with trapping them on stop codons. So then you have an even stronger data set for where's the start and the stop of a gene. And I should say that, you know, the Eugene Koonin group actually mined the ribosome profiling data to get further evidence supporting their predictions.
The second question that I had is that, so it's intriguing that sort of the small protein recruits the enzyme, which is, I think, otherwise it's not a membrane enzyme, to the membrane, and that's how it is sort of inhibiting it. But do you think that there is more to it? And because, I mean, in principle, when you're recruiting a metabolic enzyme to a membrane, you're then generating that metabolic intermediate in the context of the local membrane. So do you think that could have specific effects rather than just inhibition?
So what we speculate about is that maybe under some conditions like heat shock or low pH, the availability of NADPH or NADH, which is formed at the membrane, may be altered so that it might be an advantage under those conditions, those specific conditions, to have the enzyme closer to the membrane.
Thank you.
That's what we're speculating, without data.
Hi, Gigi. I just wanted to sort of play off of Rowan's question about stoichiometry, because it seems to me that at substoichiometric levels, one has the possibility of mosaicism of function. In other words, I'm thinking particularly of the efflux transporters, of having basically two different specificities within the same cell. And even if one were to spin this out even further, a whole spectrum of different specificities using one large framework but being modulated by different peptides. It's sort of a pipe dream, and you know it's in the pipe, but I mean, that's what's up.
No, I think it would be interesting, and I also like the idea of, can we learn more about how specificity is altered so that we could do that in a defined way?
Yes.
That
would be awesome.
I'm smoking the same thing.
Yeah, okay, fine.
I have a couple questions online. One's a two-parter, but a quick one. Is 128AA considered microprotein? And how specific is microprotein in regulating transporter activity? For example, will one microprotein only bind to a single transporter?
Yeah, so like I said at the beginning, the definition is squishy. Some groups in eukaryotes call things that are 150 amino acids microproteins. I think that's a little excessive, but it's sort of squishy. So the second question was...
are they specific? Yeah, specificity.
Yeah, we don't know that much about specificity. ACRZ is sort of curved, and that's probably what affects its specificity, but we don't know very much. The MGTS protein actually binds to two different kinds of proteins, and we don't understand that to affect transport in two different ways.
Okay, and kind of related, do neuropeptides, or are they also considered in this category of microproteins?
Again, sort of the definition, but I think there's probably a lot of cleaved peptides that have functions that we don't yet know. Mm-hmm. I guess that was sort of a point I was trying to make.
Okay, please.
Okay.
Good, go for it.
Okay, so amazing talk, Guy. So you talked about transporters and inhibition of enzymes, but are there any examples of these small proteins that activate enzymes or enzyme complexes, or independent of
Yeah.
the membrane?
So intriguingly, in both eukaryotes and prokaryotes, cytochrome oxidases have these kind of small proteins, and they increase their activity. So why cytochrome oxidases in particular might need them, or that's just where they were found. And I just want to point out for you is that they were found by crystallography.
Oh.
So in a couple of cases, people were determining structures of cytochrome oxidases, and they noticed an extra helix. So that's how the proteins were found there.
Okay. Did they like change the like how did they actually activate? Was it obvious from the structure? Or
I don't think that's clear yet. Okay. So there's speculation: are they affecting assembly, or are they? I think in the examples, it's not very clear yet.
Okay. Cool. Thank you.
Okay, maybe just repeat the CME code 66817. We've been stalling because the reception food was delivered elsewhere, but just arrived. That's why we could do these online questions. That's the secret that's out now. And but we're here. The food's here, and so let's thank you, the speaker, for our first Walls talk of this evening.
Thank you.